with us. So good afternoon to everyone. Welcome to the fifth webinar session of the CFTC summer webinars. And for today, we will have Cristóvão Dias. Cristóvão joined CFTC about 6 years ago. And since then, he has been working on the dynamics of self-organization at the microscale. He has been using colloidal particles as a prototype for the building blocks and studying how gels can be formed from the self-organization of colloidal particles. And in particular, one of the things that he's been looking at is how the mechanical properties of a gel depend on the way that the particles interact, and in particular, how it depends on the maximum number of connections that a particle can have with the neighbors. More recently, Cristóvão has been applying the same ideas and techniques to explore how to grow scaffolds for tissue engineering, trying to understand how this can be done in an effective way for growing tissues either in the lab or inside the body. And I think this is the topic of his talk today. So Cristóvão, thank you very much for your time. And also thank you for all the work organizing this webinar series.
Okay, thank you. Thank you, Nuno, for your kind introduction. So today I will be presenting a collaboration that I'm having with an experimental group of tissue engineering. So we are modeling their experiments and also use computer simulations to guide the experiments. So before we start, I'd like to say, you can see, okay, my, do you guys see my screen?
Yeah, we are seeing the first frame.
Okay,
yeah.
So, the idea of tissue engineering is to improve or replace biological tissue. So in the case of organs, for instance, there's for some time or for a long time that we can replace organs or at least use some organ donors to replace an organ. But there is some limitations. First, due to the lack of possible donors, and also due to rejection to some immune response. So one thing that we want to do, at least an objective, is to copy nature. So here I show you the example of a salamander, and these are real images. So these are real photos of the salamander growing a limb from a wound, and you can see this grow on a matter of days. So the idea is, okay, we want to copy nature. We want to at least use the own cells of the people, a person that needs the organ, to grow this organ. So there is some, we can have different possibilities, and this is already the state of the art, and some organs are already used, built in a lab, so growth in a lab. So one possibility, an organ. So we just remove a little part. So the experimentalists just remove a little part of the organ, and you grow it on a base, which is the scaffold. You can see, and then later on, it is there's a surgery that implant on the patient. This is one possibility. Another possibility is 3D printing. Now this is quite advanced, and it is state of the art also. And you can see that it is possible to print the base and the scaffold. So this is the base, the scaffold with the cells. So both the base and the cells are printed together, and later on we need to implant on a patient. So, but both these two techniques have some limitations because it's really hard to do this, to build a three-dimensional structure. So, for instance, most of these cases are used for flat organs like a meniscus or a hollow like a bladder. But is it possible also to use a dead organ from another person, but you remove all the cells, so you do the decellularization, remaining only the extracellular matrix, and this will function as the base, and then we use the own cells of the host to rebuild the organ using this dead organ or this decellularized organ as a as a as a base for the growth. This is a large advantage because the structure, which is tridimensional structures, is still the same. So there's not this difficulty of growing a 3D structure because this is already the scaffold is already created for the organ that we. From the examples that I show you, there is two things that are common to all these examples. So you need a base, a scaffold. So you always need a base for the cells to adhere and to multiply, to grow the tissue, and the organ. So you only need a base, but for all the cases that I show, you need to implant on the patient. So you need to do a surgery that can have some complications. Object, so the holy grail of this tissue engineering problems is it possible to self-assemble the scaffold for the tissue to grow. So in a way, have like in the salamander case, the organ grows by itself. Yes, using the cells of the patient as in the other case, but now inside the patient, tissue grows, and it can we don't need the expensive surgery. So this is in Aveiro, João Mano and Catarina Custódio. So they implemented a strategy where they use some colloidal particles, as you can see here, these chitosan microparticles. When they add some antibodies on the surface of the colloidal particle, that will only select the specific cells in the body that will be needed for that organ to grow, for that scaffold. So that means that we will be, the idea is to self-assemble. The scaffold will grow inside the body using the own cells of the organ that needs to be grown. Okay, so you avoid any rejection and you don't need the expensive surgeries. So as you can see, this is it's a promising idea, but it has some difficulties. Mainly, experimentally, it's very hard to try every single possibilities, the number of antibodies, the ratio of cells to particles, the densities, every single aspect you need to try experimentally is quite hard. So that's where, as a theoretical physics, we can contribute. So we can do these simulations or modeling these systems and try to narrow the range of options that the experimentalist needs to do. So what we did is particle-based simulations. I show you a schematic of how we treat the mechanisms that we consider the main mechanisms. So first we consider a spherical particle. This is a 2D scheme, but we did 3D simulation. So this is a spherical colloidal particle with some sites where. So this is the first mechanism. The cell, the free cell adheres to the empty particle, and we have this both together. We have a second mechanism that is in experiments that where cells also can adhere to each other from some clusters and on top of each other inside on the particle. And then the thing that we want is the cells can be used as a linker for particles to attach and form this structure, the 3D structure that we want. So we model this using these mechanisms, and we use molecular dynamics for the simulations. So what are molecular dynamics? So we solve the equations of Newton. So as you can see here, the part of the equations of Newton is F equal to ma. But since the particles are on to enter with the liquid part, because it's really hard to simulate all the molecules of water. We could not go to even a colloid side, or not very close to that. So if we want to build a gel or a scaffold structure with many colloids and many cells, we need to coarse grain our liquid, our medium that the colloids are. So one thing, we had these extra force terms. One is the drag term, which is opposite to the velocity. So the fluid has a counterforce to the velocity of the particles. And then we have a stochastic force. This is due to the kicks that the particle receives from the liquid. So that's why small microparticles, when you see them on a liquid, they have some kind of erratic motion, which is it's called Brownian motion. This is due to this stochastic kicks from the fluid. So this is we solve this. Then we solve the equations of motion using molecular dynamics, and we had also the interaction. We need to add the interaction between particles and between cells. So what we have here is in the term of the force, of course, and between particles and particles, the interaction is repulsive. So when particles reach each other, they are repelled. But between the cell and the site on the surface of the particle, it is attractive. So that's how we mimic the cell adhesion to the colloid, and that's how we get the linking and the cell used as a linker. We consider the cell diffusion much faster as a colloid, since the size of the cell is 10 times lower than the size of the colloid. Okay, so before we show you the results, simulation results, we need to compare with the experiments. And to compare with the experiments, we need to do an image processing of the experiments. So one of the difficulties here is because despite in experiments we don't mark the colloidal particles, they still have some fluorescence. So we needed to at least to identify the cells, identify the colloids in a way to be able to measure quantitatively the experiments. So we need to compare both qualitatively and quantitatively the simulations with the experiments. So to validate our model, we start by using some experiments. As you can see here on the top, they are the experimental results. And here on the plots, we compare simulations and experiments. Here it's the average size of cells, of clusters of cells, since we have adhesion between cells. We increase the cell to ratio, the cell to particle ratio, we increase the size of cells of clusters, which is expected. But as you can see here, the increase on the simulations and increase on experimental is quantitatively similar. We also see the distribution of the cluster size distribution of cluster cells. So we can see that in both cases, both experimental and simulations, we have an exponential distribution of cells, which at least validate our model, and then we can explore more the parameters and also try to add some contribution to the experiments to guide the experiment. So the first thing we did is propose a new mechanism. So what happened if we block the cell-cell adhesion? What could you see in the simulation? So since this is a proposed theoretical mechanism, we did the simulations in both cases. So when we block the cell-cell adhesion in the simulations and allow the cell-cell adhesions, we have on top some snapshots of the simulations. How do we do that? Instead of having an attractive interaction between cells, we just put a repulsive interaction between cells. So the attractive interaction is only between cell and site. So if one cell is adhered to the particle, an empty particle can arrive and just form a bond between particles. But if both cells are occupied, sites are occupied by cells, they have a repulsive interaction, and so the bond will not occur. So as you can expect, if you block the cell-cell adhesion, you'll have a maximum. So you will kind of occupy all the sites, so the bonding will not be allowed. You kind of poison all the possible sites to form a bond. So we'll have a maximum. But what is interesting is that adding this mechanism of blocking the cell-cell adhesion, we kind of improve the aggregation of the bonds between colloids. So the size of the cluster, the largest clusters, as you can see here, increase and is higher than the size of the on the case where we allow cell-cell adhesion, which is a little bit counterintuitive, because if we remove one mechanism of adhesion, you at least will injure the growth of the scaffold. But in this case, no, we see that it improved the scaffold formation, and so one of the explanations, the explanation that we have for this is that since the cells can adhere to each other, we'll have clusters of cells that will be formed, and these clusters of cells is basically the same as having a lot less cells to bond or to act as a linker, because one single cluster will be only one single linker between two particles. So in this way, we are actually adding these mechanisms of cell-cell adhesion. We are actually reducing the available linkers for the scaffold to form. So as you can see here in this plot, we have the cell efficiency. So it's the number of cells that are used for a bond, the ratio of cells that are used for a bond. And you can see that if we had cell-cell adhesions, is the case of the red, we have a lot less cells. Of course, if we increase the number of cells enough, since for the case of blocking the cell adhesion, we have a maximum, and then you have poisoning of the sites. There's some certain point, a certain ratio of cell to particle that we have domination between, not the mechanism of cell adhesion dominates. So this is a new mechanism that we propose, but these are simulations, and we want to, if we have a mechanism that can propose the experiment, as I say, that this is not just some thing for simulations, but it's a robust mechanism. So what we did to check if the mechanism is robust is we did a mean field approximations. So these equations, we divided our systems in 4 types of populations. This is a population dynamics, the same as you see every day on the news for epidemic models, but in this case we use it for our population of cells and sites and particles with cells and particles binded. So what we have here is we consider the sites or the patches are free. So this is a mean field approximation. In the mean field, we consider an uniform distribution. So there's no special fluctuations. There's no special correlations. We average everything. So to see if it is robust, just the mechanism, see if just the mechanism is important. So this is for the blocked cell-cell adhesion. As you can see here, we have 4 types of populations of particles. We have the free sites or free patches. We have the free cells without any bonding. We have the cells that are adhered to a particle, to a site of the particle. And then we have the cells that are used to bond to particles. So they are adhered to 2 sites at the same time. So as you can see, the dynamics is quite straightforward. We have 2 mechanisms. So the first mechanism is the mechanism of cell adhering to a site, and here it reduces the populations of free sites. It reduces the population of free cells, and it adds to the populations of cells with one site. And this has some reaction rates that are And the second mechanism is when two particles, one without any cell and the other with one cell, uh, they form a bond. And here is the second mechanism. So one patch disappears with this mechanism, another rate. So the rate is different because the diffusion coefficient of particles is different, is slower, because particles are larger. And you can see we add these mechanisms. So this is, we just solved these differential equations, and we wanted to compare with the case where we had we have cell addition. So we just add just a few terms to the equations, where in the case of the patch there's no effect, but in the case of this empty cells, we can have now one cell, empty free cell, that can go and adhere to a site that is already occupied by a cell. And you also have the case where two cells adhere to each other and form a clusters of cells. And this is the same in all cases. So you wanted to see if the mechanism is robust, and if it we can see the same behavior from the also in the simulations. So this is the bonding fraction. So this is a fraction of sites that are used to bond to two particles. And as you can see, we have the same behavior as we saw in the simulation. So we have a maximum for the case on the blocking the cell adhesion, no cell adhesion, and we have a lower bonding fraction when we add cell adhesion. So that means that this mechanism that we are proposing is very robust. If it's robust to these mean field approximations of no special correlations and no special fluctuations. We also have, at a certain point, we have a crossover. So as we saw in the simulation, there's a crossover when the mechanism of cell adhesion is more efficient. This is, as I said before, because of the poisoning of sites from cells. And above this crossover, the cell adhesion mechanism is more efficient. We test this for different values of K, so cell diffusion over particle diffusion, and we can see that even for different values of cell to particle diffusion, the crossover is more or less remains more or less the same, and which is important if we want to implement these for different types of particles and different size. So since we are interested on the build of the scaffold of this large structure, we have to see how this is affected, and where how this cluster is built, or this structure is built. So since we are a mean field approximation, we have to do some simplification. So one approximation that we can do is that consider the cluster of particles as a tree-like structures. And for a tree-like structures with 6 sites where particles can bond, we have a percolated transition. This is the same transition that Nuno told you last week, as you remember, and we have a percolation transition. And so above this percolation transition, we have gelation, as you can see here. You have gelation. Below this, we don't have gelation, so no gel is formed. So if the bonding fraction is below this line, we don't have gel, so this only have some clusters, so no scaffold is formed. The gelation is important because it's the scaffold is a gel, a structure, infinite structures that is formed. And for a high ratio, very low ratio between diffusion of particles and cells, we also have no gelation for high values, which is interesting. And I can see here is that we can kind of control the gelation transition, or if we are above or below the gelation transition, by changing the ratios of diffusion. So we can experimentally, could be done by controlling the size of the particles, or some aerodynamic radius of the particles. So we can I show you that the mechanism is robust. So the mechanism that was seen for simulations and proposed in simulations with this mean field approximation, and the mechanism is robust. So we can convince now the experimentalists to do the experiment. So we need to see if it could be observed experimentally. So the experimentalists were able to block the cell-cell adhesion. So they were able to block the ecadherins on the cells, which are responsible for the junction between cells, the adhesion between cells. And we can see, at least qualitatively, that the maximum also occurs. So this is a proof that at least our mechanism has the same behavior experimentally. Of course, the experimental blocking of the cells need to be perfect, but at least this is very promising to see that the blocking that we saw numerically in simulation, that we validate by mean field approximation, see it's very robust, and we see in simulations that the mechanism is very robust. So I would like to finish my presentation. So some conclusions. So the particle base, we see that from the first result that we were able to reproduce the cells clusters, the cell aggregation with the simulations. Knowing that we proposed a new mechanism by blocking the cell-cell adhesion, and we saw that these experiments, the experiments are in agreement with our theoretical predictions. So before finishing, I would like to acknowledge the people working in these results. So Gonçalo, that did the part of the mean field approximation for his master thesis, Nuno and Margarida, and the experimental group of Aveiro, João Mano, and Catarina Custódio. And thank you all for listening.
Okay, so thank you, Cristóvão. For those of you that want to put some questions, please use the Q&A button that you have in the bottom of your screen. And we do have already a question here. And the first question is related to the image processing of the experimental images. And the question is, how do you, if you are only considering the color, and if that is the case, how you distinguish cases where the cells are too close from each other? So it looks, according to the person that is putting
this is a very important question. So when clusters are close to each other, or at least in the same cluster, we consider the same clusters. That's what we want to check. So the first difficulty that we had is to try to measure, or at least understand on this image, what is the size of individual clusters. So what we did is to calibrate our image processing using the case with very low ratio of cells. And in this case, we have very single cells, very a lot of single cells. So size of cells. We have an interval of values because cells can spread a little bit more or less on the surface. But we are able this way to remove everything that is noise. So below a Sankey angle, and we know that above at least twice the value of the first single cell that we find, we have a cluster of cells. So here we just calibrate the size of the cells for the single cell size, and then for larger cluster or larger aggregates of colors, we have a larger number of cells. So that's how we measure the average number of cells.
Okay.
Here the challenging part is to remove the effect of the colloid colors from the experiments.
Okay, a second question is related to the experiments again, and the comparison between the experiments and the simulations. Are the experiments quasi 2D, or they are really?
In our simulations, we start by doing a 3D simulations with the patches and on all sides, and we saw the same behavior for a 3D simulation. On the experiments, what the experimentalists have is a well, so a very small centimeter size well, where we have the liquid and the colloids. These colloids are quite big, so they deposit on the bottom. So we start to have some colloids on the bottom after a few sometimes. So and then the cells that are quite are smaller, they diffuse more. So what we have is some kind of 2D like or quasi like structures of the particles, and then the cells that diffuse around. So we did that in the simulations. So we have some kind of substrate also, and we let the colloids be on the bottom of the well, as in the experiment, and then we have the cells that are diffusing around. So, but the colloids are still three-dimensional and they can rotate and everything. But we did the particle-based simulations for all case, so the case where we change the position of the sites on the particles. We consider three-dimensional without the well case, and we had the same robust mechanism. Which is interesting in this our simulations, that we are able to go to the same system size as the experiments. So we are into the same system size and time scales of the experiments, which is possible with these Langevin dynamics simulations and using a lot of the cluster of the CFTC cluster.
So that was exactly my question. So how does the number of particles compare in one case and the other in the experiments and in the simulations?
So the case that I'm using is the same range. I think even a little bit more for the simulation, but I did a finite size study and see that the mechanism is robust, and the peaks and the crossovers are not very dependent on the system size, as you can see from the mean field.
To have an idea how heavy are these simulations, can you give us an idea of how long it takes to simulate all these cases?
Okay, so I don't know, on a 16-core computer, each one of these points takes 10 times this. For 10 samples, you have to use 10 times these 16 cores computers for more than 2 weeks. So you have a few centuries of simulations here.
Okay, there is another question here that is related to the activity of the cells, in particular if the cells die or not, and if there is something that you need to consider because of that.
If the cells die?
In the process.
Okay, what I knew from the experiments is if the cells do not, during the experiment, if the cells do not have time to adhere to a structure, a surface, they die after a while. So in the case, if you don't have a space or site for the cells to adhere and grow and then get nutrients, and they will die. So what we have here that's a very interesting question, because also for the formation of the scaffold, it's important to have an idea, to be careful of the structure of that's where the simulations are very important, because if you build a structure like this of a gel-like structure, if you build it too weak, and you will not be mechanically strong enough to handle and to be stable. But if you add a lot, if you build it, if it's very dense, the nutrients will not be able to arrive to the place where the cells are, and to the bondings where the cells need to grow, and so the cells will die, as you say. So simulations, because you can have an idea what is the porosity of the scaffold to be able to get the nutrients, but also the mechanical stability. So you can also measure the mechanicals. We'll do that later. Measure the mechanical stability of these structures.
Great. So thank you, Cristóvão. For those of you that did not have the opportunity to see the previous webinars, I just want you to know that we have a YouTube channel where you can find the previous webinars, and this webinar will be uploaded there today. And if you have further questions, I believe that Cristóvão will be happy to answer your questions. You can easily contact him. And I look forward to see you in the next webinar session. So thank you very much for your time, and I'm looking forward to see you.
Thank you.